All right. So we have a cell, right? Remember this? Whoa, hey now. Someday I'll figure out how to navigate this thing. Okay. What's in a cell? PPAP. Phosphate, phosphate, adenosine, phosphate. What is that? Energy. How does we get energy? What's part of the cell? What organelle produces energy? What is the powerhouse of a cell? Mitochondria. Okay. That's become a meme lately, so that's pretty cool. Why do we need ATP? So our cells move. Okay, so our cells move. Our cells need energy. The energy is at ATP. It takes oxygen and what's the other thing you need for thinking? Glucose. If I go into a low glucose moment, I can't think. My blood sugar every so often will drop unpredictably. Don't get old, kids. If I don't maintain a snacky level of food intake, every so often my blood sugar will just drop. And all of a sudden I get ringing in my ears and I can't think. So I always have snacks. It's important to have snacks. Okay, but the problem is I eat all my snacks first thing in the morning. Could be a problem with you. Never mind. All right, so I've got a sandwich in my pocket in my raincoat, so we'll be good today. It's chicken. Should I refrigerate it? Yeah. Okay, it's cold out. Okay, so we need sugar. We need ATP. The ATP fuels the cell. We don't really care much about anything else inside the cell except the membrane. Where we care about ATP, but we care about the membrane. What's a membrane? It's a shell. It's not a shell because it's not crunchy. It's squishy. What's it made out of? Fat. Thank you. Okay, it's made out of lipids. But the lipids don't just dissolve and float away because what's stuck to them? What's a little P there? Phosphates. Phosphates form strong bonds. They require energy to break their bonds. And so by forming these strong bonds, these little phospholipid bilayer, lipid, phosphate, form a squishy membrane. There's a gap between all the phosphates. The reason there's a gap between all the phosphates is I'm going to stick proteins in those membranes. We're going to spend a lot more time talking about the proteins today in membranes. Okay, so these proteins sticking through the membranes, why are they called membrane-spanning proteins?
They're designed to go through the membrane.
Or evolved, you decide. Okay? Because they go through the membrane. They span the membrane. Proteins are made out of what flavor things? Where are the different types of proteins in here? Oh, it's written, I wrote up here somewhere. I wrote it on the board. Okay, not on here. What are the different types of proteins?
Those are flavors of acid.
That's it.
Oh, hydrophilic and hydrophobic.
Yeah. So there's hydrophilic, which means that they like being in the water. There's hydrophobic that don't like being in the water. And so if I make a chemical structure with a combination of hydrophilic and hydrophobic amino acids, I form a protein that some of it likes to be inside the membrane, some of it likes to be outside the membrane. The combination of the chemical structure forms these membrane-spanning proteins. Again, we'll talk more about those as we go on today because those become important. So I posted a picture of this. Anybody look at it? What does it look like? Is it useful? No one looked at it. Okay, fine. I'll post it again after we're done with drawing today until someone says, it's really not useful. I can't see anything. Well, fine, I won't use it then. Okay. So inside the brain, what are the basic kinds of cells? That's the next thing we talked about. It's written right there. Blood-brain barrier cells. Okay, the blood-brain barrier cells, what do they keep out of the brain?
Trash.
Well, they also keep blood cells out of the brain. There's no blood cells in your brain. They stay inside the capillaries unless you have a concussion. A good friend of ours just got a bad concussion. She fainted last night and cracked her head. Pretty good. 6 stitches and a concussion. Ouch. We're more concerned about her fainting than the concussion, because should normal adults just tip over and faint? No. So just be careful. Sometimes after a really hard workout, I feel lightheaded, but I still haven't tipped over and fainted yet. I've learned to sit down. Okay, so the blood-brain barrier cells keep oxygen and glucose flowing into the brain, and they keep the CO2 and the waste flowing out. What surrounds all of your brain cells? Cerebral spinal fluid. Good. Cerebral spinal fluid. We just remember CSF. Cerebral spinal fluid is very similar to when you have a runny nose. It's that clear, slimy fluid that drips out of your nose when you lean over. Okay, why have cerebral spinal fluid? Hello.
What you said, the base around neurons?
It's around the entire brain.
Oh.
Your brain floats in this cerebral spinal fluid. My brother-in-law, stepbrother? My dad was engaged to this woman named Irene, and we really liked the kids. They broke up and not lost their engagement, but we kept the kids, so whatever that makes them. We call him Dave. Anyway, my brother, I always call him my brother, Dave, he got hit by a car on his bicycle when he was in his late 20s. And the car hit him from behind, and he went onto the windshield and then flew forward into the car in front of him. And smashed his face into the bumper of the car in front of him. And what happened was the nose bones punched through and into his brain. If you ever watch any kung fu movies, they've got cool kung fu guys go, whoop-ah, and they kill the person by smashing their nose into the brain. Well, that actually does happen. Not necessarily with kung fu fighting, but so part of the nose membranes poke through to his brain. So for about 2 or 3 weeks after he recovered from the accident, he had a chronic cold. If you're ever in the hospital for a long time, a lot of you get colds because there's lots of diseases in the hospitals. So his nose was always running. Then he had a seizure. They went to the hospital and found out he had been leaking cerebral spinal fluid out of his nose, and he got an infection. He got meningitis from what got into his brain if fluid was leaking out. Bacteria got into his brain because fluid was leaking out. That blood-brain barrier was perforated through the membranes in the back of his face. And yeah, he recovered from that, and actually, after his accident, one eye was slightly lower than the other one, so he kind of had a halibut kind of look to him. He met this person online, and he started dating her, and we were like, dude, that's not a good idea. She also happens to have, they're very happy, been married for almost 20 years now. She's from the Ukraine, and she's an engineer. They work for General Electric in Chicago right now. He's an air engineer as well, so. Their kids' eyes are perfectly normal, though, so we're okay. We were concerned. His daughter's actually very pretty. Okay, moving on. So your brain floats inside the cerebral spinal fluid. The blood-brain barrier cells give the oxygen and glucose to the cerebral spinal fluid, and these glial cells transport it into the neurons. So your cerebral spinal fluid is really important. Has anybody ever heard of cerebral edema in here? It's a part of your brain called the choroid plexus. Not really important. It's down in the base right underneath your cerebellum, which we'll talk about in the next class. But that little part of your brain produces the fluid. What if we put too much fluid in your brain? It puts pressure. So a head injury can cause cerebral edema. When you get a blood vessel breaking in your brain, it causes the fluid to fill up with blood. And that can cause seizures and everything else. My grandmother had age, and so she started suffering from cerebral edema. Her brain was just producing too much fluid. So she went from walking to stumbling to barely able to walk to barely able to speak to becoming incontinent. And the doctors did a brain scan, and they thought it was just old people disease, Alzheimer's. And they found out, no, they put a shunt in. A little shunt, a little pressure valve right back here, had a tube that went down into her stomach. So what happened with the extra fluid? It just dripped into her stomach. The morning after the surgery, she gets up, goes to the bathroom, and comes back, and we're all like, she goes, what, can't someone take a piss? I'm like, you haven't actually got up on your own in 6 months, and you haven't had a conscious bowel movement for 6 months. But the day after the surgery, like the morning after the surgery, she was fine. Walking again, just as what she was before. So what was happening is the fluid was pushing on the neurons. And when the fluid was pushing on the neurons, what happened with the neurons functioning? It slowed down their functioning or disabled their functioning. Just that one little thing. It's pretty crazy. There's a lot of research looking into this relationship of the blood-brain barrier and the cerebrospinal fluid and long-term mental health. It's very important to exercise. Even if you hate exercising, what is exercise? How do you define exercise? Yeah. So park on the street, don't park in the parking lot. When you park in the parking lot, park on the furthest end of the parking lot, not the closest end of the parking lot. It really doesn't save much time, you don't lose that much time, and you go, well, I don't really want to walk. Well, if you exercise and you increase your vascularity in your brain, you decrease the risk of dementia by like 60%. Mentally active and physically active, you cut your risks of dementia huge. And as someone who's waiting for that in my life, because I'm almost 50, 65 to start showing signs. Okay, moving on. So it's really important to stay active. You don't have to go work out. You don't have to be CrossFit cray-cray. It's fun, but you just have to walk. Walk uphill. If you walk uphill, that's the best thing you can do for your cardiovascular fitness. Cows Mountain? Not too intense. Fury. Good. Okay. So what's the important part of the brain? No. Without the neurons, it's just a bunch of skin cells. It's just one of those old people bumps that we get. Skin tag. Ew. Anyway, so without the neurons, our brain is really just a chunk of meat. But the neurons make it special. How are the neurons make it special? Yeah. It's called an electrochemical machine. Your brain is an electrochemical machine. It uses ATP to generate electricity. The electricity is then converted to chemicals. That's what we're going to talk about today. The chemicals are converted to electricity. So far, so good? So I'm going to draw a neuron bigger, and I'm going to draw it partially on the board as well. Okay, so here is the axon hillock. So what are all neurons coated with? Nope. Membrane. Sorry, I didn't mean to sound so negative when I said nope on that. No, wrong! Jeez! Okay, sorry. So here's the soma. Here's the mitochondria. Here's the DNA. I'll label it because it doesn't look like DNA at all. Okay, kind of does in an abstract way, but. Okay, so here's the soma. At the axon hillock, we have some special structures. I'm going to erase part of that and draw this better. All right. Number one, we have what's called a pore. A pore, like pores, okay? Pores are membrane-spanning regions, membrane-spanning proteins that are little holes, and they're always open. They're membrane-spanning proteins, and they're always open. So far, so good? So I'm going to erase the connection here. Okay, so I just poke a hole in my membrane. What's going to flow back and forth across the inside and outside of a neuron? What is surrounding the entire brain? CSF. CSF is mostly water. It's mostly liquid, mostly water. The cells in there are too big to flow through the pores. So the cells inside the cerebrospinal fluid, I don't know exactly what the cells of the cerebrospinal fluid look like. Okay, moving on. I'll have to look that up. So it's mostly water. Now, inside of a neuron, the DNA produces, oh, that's cool. Okay, the DNA produces amino acids. And they're long chains of negative amino acids. A long chain of negative amino acids. So if I produce these negative amino acids, negatively charged amino acids, I collect them together, DNA makes them. What kind of things are going to be attracted to this negative charge inside the cell? Positively charged things. So things on that side of the periodic table like chlorine and bromine and iodine and etonium. Please don't eat etonium, it's poisonous. But bromine is too, but not important. But if anything on that side of the periodic table is positive, is negative, it's going to attract negative things. Anything on this side of the periodic table is positive and is going to be attracted to what? Negative things. Those things attract negative, these things are attracted to negative. So far, so good. So whatever is positively charged is going to try to flow into this pore. Following so far, so good? So I'm going to draw something which is very complicated. This is called a pump. So the way that this pump works, I'll fix this drawing a little bit. I need to put a little red dot or something on this so I can see the button on the pen better. So what this little pump does, why did I write ATP on it? It uses energy. So it uses energy, takes energy, and it kicks out sodium outside of the cell and puts potassium into the cell. So through this pore, positive things are flowing. But this pump is kicking out what? That's written up there, sorry. Sodium. Okay. Is kicking out sodium. Kicking out sodium, dumping in potassium. Kicking out sodium, dumping in potassium. So far, so good? Okay, so if we look at this periodic table, as you go down the table, the molecules get bigger. Okay? As you go down the chart, the molecules get bigger. What number is sodium? 11. It's got atomic mass of 22.9. But potassium has the atomic weight, 39? Almost 40. It's almost twice as big. Almost twice as big, not quite. So for the same mass of sodium, think of sodiums like distance runners. Little people. Okay, so we take a little distance runners versus shot putters. David? So us shot putters, do we take up more space than distance runners? Yes, so if we try to fit into an elevator, we can put more distance runners in elevators than we can shot putters. Do we still have the same charge? I don't know, I've never measured the charge of a track athlete. Rubbing balloons on them and sticking it onto things. Shot putters are generally more furry, so I'm assuming that we will have more static charge, but that's irrelevant. Okay, so if we have the equal numbers of people, who would have more mass, shot putters or distance runners? Shot putters. So what happens is, inside the cell, potassium starts building up. Potassiums are big. Sodium is being kicked out. We keep pushing the distance runners out. They're very small. So as a result, which way do you think these potassiums want to start to flow? Well, it's negatively charged inside because of the amino acids. But then something else happens. There's something called, there's electrostatic gradient. Electrostatic pressure and concentration. I totally dig the rainbow font. Sorry. And it's crayon textured. That's my favorite part. Okay, sorry. Is it too hard to read or just my handwriting? Just the handwriting. Okay, I can just ignore that one. Okay. So electrostatic pressure is the force of the electrical charge being pulled back and forth. That's the electrostatic pressure. Concentration pressure is, or it's called the concentration gradient, is the fact that if I put a bunch of potassiums on the inside and pumping them in, what is potassium going to want to do? So maybe use a disgusting example, because I will. So let's say David's sitting back there and he's had something, we ate a bunch of hummus yesterday, so your stomach's all gurgly. And he has a little sneaky gas out there. What happens to all those gas molecules? They disperse. Do they equally disperse across the room? Yeah. If you're in a liquid, no, bubbles. No, sorry, it's not the word I'm looking for. Take a drop of food coloring and drop it into your swimming pool. My kids have done this before. Take a drop of food coloring, put it in the swimming pool. You see this little glob of color sit in the swimming pool. But then what happens to that little glob of color? It disappears. Well, it's not really disappeared. Those little molecules are still in there. But what's happening to them? They become equally dispersed inside the liquid. That's a concentration gradient. Each molecule repels molecules of its own kind. And at standard temperature and pressure, molecules will find a balance of space between them. So it's concentration pressure. Which is stronger, electropressure or concentration pressure? Electrostatic pressure is much stronger than concentration. So if I have a fan in my office that has this electric plate on it. I should clean it at some point in its life, but I haven't. But what it does is it creates an electrostatic charge across this metal plate. As the air is blowing across this, what does it collect? Dust. What kind of dust? If it's got an electric charge on it, let's say it becomes negative, what molecules does it attract? Positively charged molecules stick to it. And if they run a positive charge through it, negatively charged molecules will stick to it. And so they run the two plates side by side, and it picks up all of these charged particles in the air. Deionizes the air is what it's called. The sad thing is, do you think it really does much? No, because there's so many free ions in the world, it just collects the dust that's floating by it in that little space, and the rest of it still circulates around. And all you do is get all the dust inundated on you when you clean the thing off. Yeah. It does work for air purification in a very closed environment, but not at a big campus like this. So the electrostatic pressure is stronger than the concentration pressure. So in this pore, what's flowing back and forth all the time? No, the K is potassium. Potassium. Potassium is constantly flowing back and forth, back and forth, back and forth. This pore only is permeable. So there's a word called selective permeability. When something is permeable, hopefully you're not wearing a permeable sweatshirt today. You're wearing an absorbent sweatshirt. What'll happen if you get it wet? Water soaks in. Yeah, yours is the same way. My daughter refuses to wear, she's wearing her beach volleyball sweatshirt today, walking in the rain, which is extra thick and extra absorbent, which means She'll be damp most of the day today. But she refuses to wear something which is selectively permeable. I'm a very cheap person, and I hated the fact that my wife bought this for me because it was very expensive. It's awesome. It's made out of a fabric called Gore-Tex, which is selectively permeable. Water doesn't go through it. But what does? How? I don't understand chemistry, how they make that. Yeah, it breathes. So my wife actually stole it and went running in this morning in it. And even though she's running, was she overheated? No. What if you just wore a completely non-permeable jacket? Like you see, has anybody ever worn the old plastic poncho you get at SeaWorld? Oh, bad. Because you need that air getting to your skin to prevent your skin from overheating. Or if you've ever had to walk across a long beach and you forgot to put your wetsuit on in the car. By the time you get to the ocean, you're like, you're trying to let some air in there. Because is a wetsuit permeable? No, it's only permeable on the top and the bottom. The suit itself is not permeable. So it keeps all of your body heat in. So your membrane is like a Gore-Tex jacket. That jacket only allows air to go back and forth, but doesn't allow water to go back and forth. In this particular case, what does the pore allow? Potassium to go back and forth. So there's a mathematical equation called the Nernst equation, which is some biophysicists figured out that if I make a membrane that is only permeable to potassium, okay, only permeable to potassium, I'm going to draw this little thing up here. I'm going to get rid of the rainbow font, sorry. We can add a pen. How do I change the color? I don't want the black pen. Let's change the color of the black pen to white. Bigger. Okay. So, go away, pen. If I draw an electrical current between the outside and the inside, so that's a little meter, it looks like this. Here's 0 millivolts. What charge are these amino acids? Negative. So a membrane that's permeable to potassium has a charge of negative 70 millivolts. The negative 70 means that the potassium is flowing which way? In or out? Out. Positive going out means negative. So the electrostatic gradient is causing potassium to try to be drawn in there. It's also trying to cause sodium to be drawn in there. But what do I keep doing with sodium? I'm trying to move it down. I can't move it down. Sodium is being pumped out. If sodium is being pumped out and potassium is being pumped in, but potassium keeps flowing out, we have a net charge of, say it, negative 70 millivolts. This is called resting potential. This is a lot of work. We haven't even got to 1 neuron at 1 millisecond of your behavior yet. But you see why the vocabulary is so annoying at the beginning of this class? You have to learn a lot of vocabulary just to understand something really basic. But once you get through this, then it gets a lot better. Okay, so a neuron sitting at rest has a potential energy. What is potential energy? I think I have yeah, this one works. What is potential energy? You're not living up to your potential? So if I throw this down, is this storing potential energy? Can you hear it spinning? Come on. So what it can do is if I throw it down, the harder I throw it down, the faster it comes back up because I'm putting more energy into the system. The more energy is into the system, the faster it comes back up. Okay, and so the harder I throw this down, the more energy it has. The more potential energy it has. What happens if I just leave it down here, though? And eventually it'll stop because, well, the laws of physics don't like things producing potential energy. So in order for me to keep it going, what do I have to keep doing? I have to keep giving it more energy. So a neuron has a potential energy. Its potential energy is that pump. That pump is constantly running. It's pushing potassium in. Potassium's going in, but then potassium's flowing out. Potassium flows in, potassium flows out. Potassium flows in, potassium flows out. And the potassium is just constantly flowing back and forth, except sodium isn't. Sodium is just sitting there. All the little distance runners are just sitting there on the outside wanting to get in. How come they can't get in?
Semi-permeable.
It's a selectively permeable membrane. It's only allowing certain things to go through. Now, oxygen goes back and forth across. No problem. CO2 goes back and forth across. No problem. The cell produces some extra CO2, needs some oxygen. Glucose is actively transported into the cell. But glucose, if you mix sugar and water, does it actually create an electrical charge? No. If you shake up your water bottle and add extra oxygen to it, does it change its charge? No. It's O2. Anyway. So what's interesting about this is that these pores and these pumps maintain this resting potential. Maintain the idea that this cell has the ability to do work. So far, so good? So we haven't even gotten to one set of cell behavior yet. Okay, so what I'm going to do is I'm going to draw this again over here, even bigger. Here's the axon hillock again. Okay, let me draw the pore here in the green so you can see the pore. There's the pore. I'm going to draw this other one in white, and it's going to be a gate. Now, a gate is a membrane-spanning protein. It's a membrane-spanning protein, except how is a gate different than a pore? Gates open and close. Wow. There's different kinds of gates. Okay? There's voltage gates. Guess what opens a voltage gate? Voltage. There's chemical gates. Guess what opens chemical gates? They're called ligand, actually, but what opens a chemical gate? Chemicals. There's mechanical gates. Guess what opens a mechanical gate? It made the V into a 0. So it turns out if it auto-shapes, it doesn't like to erase. So there we go. Thank you.
Modern problems require modern solutions.
Yep. Uh-oh. I'm writing the words up here that you will see. Voltage activated gates are activated by voltage. Chemically activated gates are opened by ligands or chemicals. Ligands is a name for chemical. Mechanical activated gates are opened by movement. So if you run your fingers back and forth across your piece of paper and run your fingers back and forth across the little bumpy, annoying desks that you try to write on, can you feel the difference between the desk and your paper? Do you think that's voltage, ligand, or movement? Say it louder. Movement. So what you're doing is you have types of receptors in your fingertips, we'll learn about these later in the semester, called Pacey and corpuscles, that all they do is measure movement. If I take a stick and poke that neuron, when I poke it, the neuron bends like a chapped lip. And when it bends, guess what it lets in? So what if I open this neuron? What's a lot on the outside? What's a lot on the inside? Potassium. So what if I open a gate? What's going to happen? I wanted to erase that. There it goes. If I open this gate up, which way is sodium going to flow? Why in? That's right. But so why does sodium want to come in? Because it's negatively charged inside, it's positively charged on the outside. Also, what have we been doing this entire time with the pump?
You've been pumping it out.
I've been pushing it out. Chemistry does not like it when you force one thing to go to one direction. What happens as soon as I turn off the electricity with these lights? They turn off, but the light works by running electrical current through the gas. The gas gets excited, the gas moves very quickly, and releases energy. But then when I shut it off, the gas slows down and stops moving. And when you first shut, if you ever sit in a really dark room, you shut off a incandescent, one of these, not incandescent, these are fluorescent tube lights. The fluorescent tube lights they turn blue. And then they'll sit blue for a while before they finally fade away. If you're ever having fun in the dark, take a fluorescent bulb and scuff your feet on the floor. What can you get the light to do? Flicker. Next time it's at Santa Ana, grab a fluorescent tube and go walk around the library at dark. Okay, that's so weird. Don't do that. Just drag your foot around the rug, and you can actually light up a fluorescent tube just by dragging your foot around. Because the electricity goes in and excites the thing, the molecules. The molecules move around for a little while, and then they use the energy, and then they stop moving, and then the light goes away. Obviously, you're not 12, because that's totally cool for a 12-year-old to do. Not that we've done that at our house at all. The small little incandescent bulbs that you put on your lamps, you can totally light those up just by dragging your feet around the rug. Okay, fine. So, the pumps are dumping sodium out. If I open a voltage-activated gate, what's going to come flooding in? Sodium. Because sodium is being pushed out and it's negatively charged inside. Yeah.
So you're saying sodium is going to flow
Into
from the outside?
from the outside, because the pump is taking it from the inside and pumping it to the outside. Pumping it out, pumping it out, pumping it out. Get out, get out, get out, get out. As soon as we open that gate, there's a rush to come in because that pressure is built up. The easiest way to think about it is when you're scuffing your feet around and you see someone who has an earring on the top of their ear and you're like, oh, zap. Okay, how long does the spark last? It'd be really cool if you go like, you just really cook them. But it doesn't, because does nature like to have this imbalanced charge? No, so the spark only lasts a very short period of time. In a real static spark, very fast, 100th of a second. Some lightning strikes are 10,000ths of a second. Boom. That on last week, I forget what day of the week. What day did we have the bad thunderstorm last week? Thursday. I was sitting here waiting for the kids at school, and the lightning and thunder were simultaneously at my son's school. A bunch of pictures fell off the wall in my son's classroom. It was really cool. And I'm sitting in my car, and I jumped. And I saw a couple lightning bolts. I'm like, yeah, cool, lightning. I like lightning. And my friend who's sitting in the car in front of me texted me, holy shit. And I said, it's the ghost light. That's my standard response to anything. If you're a Mater fan, that's funny otherwise. Never mind. Okay. So who cares that sodium comes rushing into the cell? I'm going to go back over here to my little voltage meter. If I dump sodium into the cell, what's going to happen to the charge? It spikes. The charge goes all the way up to plus 30. Spark, does it last very long? No. Well, the pumps are relative. Mechanical things are much slower than electrical things. So mechanical things, that's why I love watching old people use cell phones, because they're using them like a mechanical device. because they're trying, I don't want to break it. There's no moving parts. Unless you drop it, it ain't going to break. But they're using it as if it's a mechanical device. It's entertaining to me, at least. So, yeah,
You said it was plus 30.
plus 30 millivolt charge. So what happens, a cell actually flips charges. Boop. For a split second. I saw that. Okay. Then the cell rebounds. Well, that's important. It rebounds. So this whole process right here that we're talking about, it's about 1 to 2 milliseconds long. Spark. Yeah.
Is the plus 30, like, is that the max that it can be, or is that just an arbitrary number?
Okay, so the answer is it's not an arbitrary number, but it's not the max that it can be. So there's afterwards she asked me some questions in the office, so I showed her my Principles of Neuroscience book. Okay, in the Principles of Neuroscience, it's like the giant book of this class. Like if you ever want to go to graduate school and study this stuff, you buy the Candle and Schwartz Neuroscience book. The reason why it's cool, hold on. Okay, here's the whole thing. Okay, this book was made back before there was 619s, because I wrote my friend's phone numbers in my class. Everything was 619, so we just wrote the phone number down. This is 1991 or 1994. I forget exactly what edition this was. Oh, you're back. Okay, this is not with reading glasses on, 1991. Okay? In the class right now, in here, did I drop that little index card that was in here? We are on this chapter right here. If you really want to look at this, but there's these mathematical formulas in here that tell you why it's plus 30 or minus 70 millivolts. Some biophysicists figured it out. For psych majors, we just go, okay. It's exciting to know. But like, if I talk about membrane-spanning proteins, there's a whole chapter about the DNA sequences of membrane-spanning proteins and the entire genetic code to create a potassium pore or a sodium channel. It's all in here if you want to look for it. Why don't we really need to know that? You need to be able to take the basic ideas of this and apply it to things that are important to most people in psych. Well, okay, so what if I give you a little alcohol? Aside from getting fired, what would happen? Giving you ecstasy last class, come on, ecstasy, no problem. Well, so alcohol, or the drugs that William takes for his, I did that last semester too, just for fun, takes for his epilepsy, lowers his membrane's potentials to minus 90 millivolts. If you drink alcohol, you lower your membrane's potentials. Does alcohol care which neurons it's affecting? No. It slows them all down. What if you drink even more? Well, eventually you suppress enough neurons to lose consciousness. If you lose enough neurons to lose consciousness, is that dangerous? Yes, because right below consciousness is respiration. That's important. A lot of people that get really hammered have shallow respiration. They can't seem to catch their breath. That's because their brain's not breathing at the right rate. They're breathing slower than their body oxygen levels need. So you feel fine, but well, you're dying. Not a good idea. So let's go back to the bigger picture, and this is why I want to kind of pay attention to this. During an action potential, there is a flow of neurons back and forth across, where are we in the cell? Not neurons, a flow of ions back and forth across what part of the cell? The axon hillock. We haven't even gotten to an axon yet. This is in the soma. The axon hillock is right there, and we have a flow of ions here. If the flow of ions is enough, it opens up these gates. When it opens the gates, we get an action potential. Okay? So what flows in if I open this gate? Sodium. So far, so good? Sodium flows in. So I'm going to keep drawing this neuron out as we go here. So my neuron's going to go, and my neuron is now I'm into the axon. So concentration gradient-wise, which way is the sodium going to flow? So if I dump a bunch of sodiums in here, which way are they going to go?
They're gonna go away from the potassium since they're positively charged.
Oh, I thought I could do it. Okay. That would have been cool. Okay. Oh, I can't get rid of that dot. Anyway, so the sodiums move this way, and the sodiums move this way. If they dump a bunch of sodium into the water, it's going to spread out equally amongst the cell until eventually there's no concentration anymore. It's equally concentrated. Following this so far? Well, it turns out that as the sodium moves throughout the cell, sitting right here is another voltage gate. If I bring sodium over here, what's going to happen with this gate? What does that mean? Thank you. I saw this. Okay, if this opens, what's going to flow in here? More sodium. And I'll draw the graph here below this so you can start to see. But if sodium flows in here, which way is sodium going to flow? It's going to disperse, but which way is it going to go? There's already a bunch of sodium up here because it flowed in back there. So it's going to go, well, that way. I'm looking at my computer going this way. It's going to go this way. So the sodium's going to flow back this way. And it's going to keep going until it finds a gate. And when it finds another gate, it's going to keep flowing this way. How come it's not going to flow backwards? Because the concentration gradient pushes it away. So it gets salty here, and then as it gets salty here, this part of the cell lets more sodium in. Gets salty here, but it's already salty here, so it's going to go away from it. And so we have this little like whoosh. This little whoosh that goes away from the axon hillock. It starts at the axon hillock, and I'll show you why it starts at the axon hillock in just a few minutes. But it starts at the axon hillock and then slowly moves down the axon. If the axon branches, and now there's two branches of this, there's two sprouts now, each of them with sodium channels on it. What happens at each one of the branches? Sodium comes in on each one of the branches. And so as I keep going, see, I could never do this with the whiteboard. But as I keep, and you can't draw it potentially at all. Anyway, not important. So as it keeps going down the pipe, if this one branches, and I just put some sodium channels on here, what is sent down each of the branches? This surge of sodium is being sent down to all the ends of all the neurons. Just picture that neuron from the connectome video, how many different little ends and connections it has. But all I have to do is send from the axon hillock at the top of the cell, send the signal. Does it go all the way to the end of all the axons? Yeah. It's called the all or none law. I'm just going to go back to this. Let me shrink it down a little bit. So in my neuron, the contrast ratio sucks, so we can make it a little bit bigger. In my neuron, I have lots and lots of sodium channels. These are actually called nodes. They're called nodes. So wherever there's a node, what is there a cluster of? Sodium what? Sodium channels. What kind of channels are they? They're voltage activated sodium channels. So following this, accent hillock, lots of voltage activated sodium channels. Voltage activated sodium channels open up when there's a positive charge. Voltage activated. Sodium flows in. Sodium is positive. So what does it do to the next node? Positive makes that channel open, which goes to the next node, which causes that channel to open. So across down the entire neuron, we have action potential, action potential, action potential, action potential, and as it goes down, no matter how many times it spreads out in branches, what's it doing at every single branch and every single sprout? Sending more action potentials.
So as you get more and more sodium in, is it actually increasing the charge or keeping the charge?
The charge is based on that formula in the stupid book. So the charge always goes up to plus 30, and then rebounds to minus 90, and then goes back up to norm resting again.
When does it rebound?
Well, that's actually the difficult question. When does it actually rebound? So I want to draw one more thing on here, just to confiscate this as much as possible. I really need a yellow, so I'm going to get rid of the second blue one here. And I want kind of a, beigey yellow color. That's good. Make that a highlighter beigey yellow color. Can I change the color of the highlighter? No, I can't. Oh well. So I'm going to draw this right here. One of these days I'll get some more pens for this and I'll figure out exactly how to do this. So I drew this kind of beigey yellow color. Thank you, that actually makes sense. This is fat. Back when in F, 416, sometimes I teach in that classroom over there. They teach one of the health classrooms. They have this cube of fat. It's a 5-pound block of fat. You know, it's supposed to be part of like your health thing, and they have this big block of, here's 5 pounds of fat. Anyway, but, so I try to use that fat color. This is fat, it's myelin. In our brain, it's white, not gray, or not yellow, but I always think of yellowy fat, something, okay? But so this is fat, this is called myelin. Myelin coats the neuron between the nodes. So in between the nodes, there is a glob of myelin. What that myelin does is allows the neurons to send signals with less interference. Baby neurons don't have a lot of myelin. As a result, baby behavior is extremely slow. Kids don't fully myelinate their brain until 4 or 5 years old. So like when you talk to a 2-year-old, you don't have to use baby talk, but should you pause between words? Notebook. 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 Why is Blue's Clues and Dora the Explorer so freaking irritating for adults? It's too slow, but for a child's brain that's not myelinated, right on track. Boots. Did you see Swiper? Yeah, he's behind the rock. Swiper, no swiping. Kids, can you say Swiper, no swiping? Kids are like, Swiper, no swiping! You know, in the real world, he would have been dead by now. But that's not important. Swiper would have eaten that damn monkey at this point. Okay? Sorry, a little rage for Dora. Okay? But the reason why those shows are so painfully slow is because kids' brains are not myelinated yet. So their neurons require significantly more of these little action potentials just to get the signal to the end. But as you get older, guess what we do with our myelin? No, we myelinate not too much. We myelinate more. We think faster. In other words, to have the same thought, this Dr. Riley, the old physics professor, I love him dearly. I asked him a question a couple years ago before he retired. I go, okay, are you better at solving problems now compared to when you graduated college? And so his answer was, which problems are they? The problems that he teaches all the time, the mathematical stuff that he teaches in his physics classes, he can solve so fast because those pathways are really heavily myelinated. It requires significantly less energy to have the same solution to a problem he's solved 1,000 times. What happens when he tries to solve a new problem? His pathways are so myelinated that they don't branch. Because wherever there's myelin, is there going to be a sprout right here? No. That means that that neuron is sending the signal to someplace. Wherever it gets to dendrites is when it branches out. And this is why a lot of old people have the same story for everything. I remember when I was your age, we've heard this story before. My father-in-law loves telling the story of him and his friend Dean, Dean and his now wife, but then girlfriend, were smooching out in Mission Trails Park, and him and his buddy threw dirt clods at their car. And so he chased them and then got back and stole the distributor out of their car so they couldn't drive home. And I've heard this story so many times. Why does he keep coming back to that story? It was really funny, because then they stole a distributor out of an old pickup truck at the gas station by Patrick Henry and used that in his car so they could get home. And the next day brought it back and left it on the hood of the pickup truck at the gas station. You know, a little car repair place right by the football field with Patrick Henry? That was a gas station. And so what's weird about it is that he tells that story to us all the time as if we've never heard it. You're old, Grandpa. Because when you remember something, that's activating a neural pathway. But the more you myelinate your connectome, what happens to it? Ooh, I would say that the pathway becomes unchangeable. Old dog, new tricks. It's harder to teach old dog new tricks because they know what they know, and it's hard to change what they know because of the myelination. That's why early education is so important for little kids. Okay. You feeling okay so far? Not too bad? So here's how this looks on a, use a darker color here. Here's how this looks on a chart for your electrical chart. Here's 0 millivolts. Ready for this? If I let some ions, some sodium into the cell here, what's going to happen to the membrane potential at this node? Sodium's going to flow in and go all the way up to plus 30. So at plus 30 millivolts, what is running away at plus 30 millivolts? Inside the cell, what's running away? Why is the sodium running away on the inside at plus 30? Not just concentration, but also dispersion because of the electrostatic pressure. If all of a sudden all the sodium rushes in and it's super positive right here, they're gonna go push either way. Okay, so now I'm going to tell you something that we'll get to next class, but if it's high sodium in the soma, that's what's going to cause the axon hillock to fire. If it's high sodium in the soma, that's going to cause the axon hillock to fire. So as a result, the sodium flows down through this myelinated mess. Okay? And so I don't know if I can erase part of this. We'll find out. Oh, well, that's a mess. Okay. Sorry. Okay, so I'm just going to draw a little gap in the myelin here so I can put a node here. So as the sodium flows in here, sodium flows down, this cell, what's called, hyperpolarizes. It goes negative. So this part of the cell, once it goes positive, all the sodiums run away, and it becomes negative, super negative. So the second part right here, I'm going to draw this in a different color. When the sodium is at its peak, guess what this next node is going to do? it's going to fire. And then it's going to rebound. I'm going to change this one to red. Come on. All right. So when this one, right, let's put this one right here. The red doesn't work very well, but whatever. When this one is, when the second one is at its peak, it causes the third one to go up. Let me make that even bolder. And when it recovers, what's happening with the next one? The next one starts to increase. So what you're seeing, kind of works, not exactly right, but what you're seeing is as the first node depolarizes, becomes positive, it causes the second node to become positive. What's happening when it becomes positive? Sodium's flowing in. When the sodium flows in, it doesn't go back, it goes down, which causes the next one to fire. After every fire, however, after every electrical signal, the cell resets itself. So when it goes negative, guess what some of the sodium does? What would some of the sodium do if this is flowing in here and this is negative? It's going to flow back. So you have this whoosh, whoosh. Picture a throbbing headache. You know, ever get a throbbing headache? What do you feel? It's not your pulse, actually. If you actually measure your pulse and measure your throbbing headache, they're at a different frequency. Usually not really paying that much attention when you have a throbbing headache because you have a freaking throbbing headache. What you actually feel is your brain contracting and swelling. Your brain has its own pulsating rhythm. It's really irrelevant to neurons, but it's a nice visual. That if you actually look at a brain during a surgery, it goes whoosh, 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 whoosh. And it circulates cerebrospinal fluid through this contraction and constriction of the membranes around the brain. When you go into deep sleep, your brain cells shrink. Whoosh. And it floods the brain with all the wastes and things into your blood system. And you flush out all the nasty chemicals in your brain when you're sleeping. Is sleep good for you? Yes. Very good for you. Okay. So, again, how long does each one of these little spikes take? 1 to 2 milliseconds each. 1 to 2 milliseconds each. They're affected by what neurotransmitters? Sodium. Not transmitters, I'm sorry. These are affected by sodium. Sodium is the primary mitigator here. Well, next class we're going to talk about how potassium is involved as well. Because potassium is involved with this as well. Potassium flows back and forth through the pores and through other channels, but sodium is the key. What keeps the cell at minus 90 or minus 70 millivolts? Let's go back to the beginning. What keeps the cell at minus? Come on, get bigger. There you go. What keeps it back here? Life was so much easier back here. Why won't let me zoom in, you stupid thing? That worked, whatever I just did. I think it's just random. I think it's messing with me. Okay. What keeps it at minus 70? Pump. If I disable the pump, what happens to the neuron? Doesn't die right away. It's not fatalistic. But this has all happened to us. My friend that passed out, if you lose oxygen to your brain, you pass out. How fast does your brain stop sending signals? Very fast. How long do you have when you start losing oxygen? 3 or 4 seconds. Yeah, it's great. 3 or 4 seconds, you're like, oh no. That's the problem is if you lay in the sun Can you stand up too quick? Where does all the blood go? Right to your feet.
But for a brain, 34 seconds is like an eternity,
Oh yeah, because without that oxygen, your brain can't maintain conscious functioning. Pretty crazy. So it's amazing how fast if this ATP is stopped in your consciousness in your brain, it's amazing how fast you lose brain functioning. I remember my friend was a pilot and he was flying around. We were flying from here to San Jose because he wanted to go on a long distance trip. We had to go at Bakersfield Airport or something. We were stopping to refuel, and we had to go up to a certain altitude to get past thunderstorm some other air traffic was coming in. We had to go up to 15,000 feet. We didn't have oxygen. And so you could stay up there for a certain period of time before you had to come back down and re-oxygenate yourself. And so I didn't know it, but my brother who was flying the plane goes, yeah, you were singing the whole time. I was? Because my fingernails turned blue. So what does that mean? My oxygen saturation level went down to about 75, 80%. And so I was, I sang it away. And he's like, yeah, Tower, we need to go back down. My co-pilot here has gone cray-cray because he can't breathe enough. And thanks for that. But I've also passed out at altitude before. So for me, if I go up to high altitude, do I have to be careful? Yeah, other people survive high altitude really well. I don't. Maybe it's because, yeah, if I'm up in the mountains for a couple days, I'm fine. But like the first day up there, if I go too hard, I'll get nauseous or migraines or whatever else. And that's literally because my body's hematocrit, the blood level, can't maintain enough oxygen, and what happens to brain functioning? Almost immediately. Doesn't cease. That would be bad. But it decreases almost immediately because you need to have this. You need to have this action potentials. Okay, I know this is not the fun stuff in the class, but after watching the Connectome video, if you haven't watched the Connectome video, look at the announcements in Canvas. But you start to realize that each cell is sending its own electrical signals. Are they completely all intertwined with each other? Yeah, so next class, we're going to take apart the synapse, and we're going to take apart how they actually communicate to each other. Yay? All right.